between the first neuron and the second neuron. So the electrical impulse can directly pass through the connection from one neuron to the another neuron. Whereas in earlier case, chemical synapse, there is a gap between one neuron and the other neuron. We will discuss more about a synapse in coming sessions. In the session 1, we discussed about what is a synapse. Synapse is a tiny space between two neurons, right? So if you have a look at the picture, this is the presynaptic neuron and this is the postsynaptic neuron, right? Both cases, this neuron as well as this neuron are presynaptic, this one and this one are postsynaptic neurons. That means this is the first neuron, let's consider this is the first neuron, this is the second neuron, first one, second one. So signal always transfer from the presynaptic neuron to the postsynaptic neuron, right? So whenever action potential reaches to the terminal buttons, axon, we discussed about axon terminal buttons. So this is the axon terminal button. So whenever the signal action potential reaches here to this particular point, so the calcium signaling helps in vesicular transport of the neurotransmitter, and the neurotransmitters will be undergoing exocytosis and then they will be released into the synaptic cleft. So this space is called synaptic cleft, and the neurotransmitters like acetylcholine or GABA or dopamine will be released into the synaptic cleft, and then this neurotransmitters will move through the synapse and then it will bind to the receptors which are present inside the postsynaptic neuron, right? So this is not a random binding at receptors, only a specific ligand can bind to a specific receptor. So this is GPCR, G protein coupled receptors, this one, and these are acetylcholine receptors. So acetylcholines can only bind here to this particular type of receptors, right? And then once the signal is passed here, from here to here, the action potential continues here in the second neuron, and then it will move on to the third neuron, and then it will move on to the fourth neuron. Likewise, the signal will continue from one neuron to the other neuron, right? So this is about chemical synapse, and what you see here is electrical synapse. So in case of electrical synapse, so there is no space in between the one neuron and another neuron, and there is a direct connection between first neuron and the second neuron. So the electrical signal will directly move through these particular channels, the proteins which are integrin proteins which are present in between the two neurons, and then the electrical signal will pass through these molecules which are there in between these two neurons. So this is about chemical synapse and electrical synapse. So few important terminologies which I discussed just now. To summarize, presynaptic terminal, that means presynaptic neuron axon cleft is called a presynaptic terminal, which is going to release the neurotransmitter, and the synaptic cleft is nothing but the synaptic space which is there, and the postsynaptic membrane present in the postsynaptic neuron, that is in the second neuron, which has the receptors for capturing the ligands. Then other important terminologies like your neurotransmitters, important neurotransmitters like acetylcholine, dopamine, serotonin and glutamate. These are the important neurotransmitter released from the presynaptic neuron, going and binding to the receptors on the postsynaptic neuron. Synaptic vesicles nothing but a small tiny sacs which are released by the presynaptic neuron. These sacs will be carrying the neurotransmitter, and then they will be releasing this into the space synaptic cleft with the help of exocytosis. And receptor protein, so these receptors are generally present on the postsynaptic neuron, like we have seen GPCRs and N-acetylcholine receptors and other receptors specific for ligand binding, neurotransmitter binding. and neurotransmitter release also from the presynaptic neuron happens only when there is a signaling, only that is done with the help of voltage gated calcium channels. And next one, signal transmission. So whenever the neurotransmitter comes and bind to the postsynaptic neuron, the signal will be propagated from one neuron to the another neuron. So once the signal is transferred, whatever left out neurotransmitters present in the synaptic cleft will be reuptaken into the presynaptic neuron, and also they will be degraded, right? So these are the important terminologies in case of synapse, right? So you can see the pictures, and this is a chemical synapse where a presynaptic neuron and a postsynaptic neuron is there. You have the vesicles, you have the release of vesicles by exocytosis, and the neurotransmitter moving and binding on to the receptors present on the postsynaptic neuron, and then the signal is propagating from one neuron to the other neuron. So you see very tiny space between the two neurons, right? So this is about the chemical synapse, generally present inside the brain in the gap junctions. So there is a direct connection between one neuron and the other neuron. If you consider this one as a presynaptic and this is a postsynaptic, there is a direct connection and the electrical signal will be propagated here. IP3 and calcium can also move through these receptors. So this is about the session CO1. In 2 minutes I will start the next session. Thank you.